wanted to give you a little bit more molecular detail on the structures that we showed in the previous webcast. You do not necessarily have to know how to draw these, but it should make more sense to you if you can see what's going on here. The sugars, the two sugars we have are ribose, and we have deoxyribose. And as the name would imply here, the deoxy is going to be missing an oxygen. So these are just regular old pentose sugars here. So I'm going to cross those intersections. This would be carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. And of course we add the primes because we also have the bases are numbered here. We have the beta position here, which means that we're up in the number 1 prime molecule. We have hydroxyl in the 2 prime position, hydroxyl in 3 prime position, and a hydroxyl in the 5 prime. Now, interestingly enough, what's going to happen by the time we get finished is this hydroxyl will actually be linked to the nitrogen base. This hydroxyl right here will be free. This hydroxyl will be linked in the phosphodiester linkage, and so will this hydroxyl in the phosphodiester linkage. Let's look just at deoxyribose. I'm just going to copy this and move it over here so you can see it's identical except for one thing, and that is that we're going to be missing the oxygen down here in the number 2 prime position. So again, this is ribose with the hydroxyl in the 2 prime position, and deoxyribose, which is found in DNA, with the hydrogen in the 2 prime position. Okay, so there's a little bit more on the sugars. All right, so now let's look at the bases for a second. The bases occur in 2 flavors here. Again, and I'm not putting a detail into this because I just want you to see the shape here. So we have the pyrimidine base, which looks like this, and we also have the purine base, which is similar, has the same basic structure. No pun intended. Here I'm going to put nitrogen there and a nitrogen here, but then there's an additional 5-membered ring attached to the 6-membered ring. Now, what we're interested in here specifically in terms of making this molecule is we're interested in the fact that there's a hydrogen hanging off right here, and then there's also a hydrogen hanging off right here. And it's these hydrogens that are going to be used to connect the sugar to the base. So let me go ahead and just paste back in one of these sugars that we did before. Let's grab, for instance, ribose here. So let's paste in one of the sugars that we had before. And so hopefully what you can see immediately is that we're going to be able to have a condensation reaction right there. So we'll pull the water off of here, and then you can see that we would have this first linkage, which would connect the sugar to the base. So there's one linkage. The same thing had happened here, except we would just attach the sugar right here to the number 9 nitrogen instead of the number 1 nitrogen over here. So that's how that hooks up. All right, so now what we're going to do is we're going to take this structure right here, and now we want to add a phosphate group. If you recall, the phosphate group actually was a phosphorus. It has a double bond oxygen. It has a hydroxyl here. It has an O minus here, and we'll put a hydroxyl right here. And so what we're going to do now is we're going to actually connect the phosphate to the alcohol. So this is an organic acid to an alcohol here. And again, what I'll do is I'll do a condensation reaction like this, which will produce a water molecule here. So when I do that, take this out right here. So now we'll have connected the phosphate directly to the sugar. So again, what we did was we started with the sugar, we added the base, and then we added the phosphate to the base. So this is actually the structure here. It kind of spread out a little bit because of the length of the bonds, but this structure right here is actually the nucleotide. And by the way, if I want to make this a nucleotide containing a purine base instead of pyrimidine, I just come up here and change this right here to the purine base. And if I want to make this a DNA nucleotide, I change that hydroxyl right down there to a hydrogen. All right, so now let's take that's your monomer there, which is kind of large. Let's take that monomer and actually put it together. So what I've done, I've copied it and reduced its size here. Let me clone this for a second. We'll clone it. So now I've got 2 of them. So what's going to happen now is I'm going to bring this guy in here like this, and I will make my next phosphodiester linkage will be right there, or my next ester linkage. So if we do this yet again, so if I paste another one, you can see I'm simply going to continue to add these guys one after the other, and as I do that, I'll actually will get my tetranucleotide in this particular case. And the only thing that's different in each of these is which bases go here, and there's nothing that restricts which base goes where. So what I've done here now is I've gone in and I've established all these last ester linkages here, one there, one there, and one there. And what you can see now is that if we look at the hydroxyls, the 3' and 5' hydroxyls, you'll notice that only this 3' hydroxyl is not involved in a phosphodiester linkage, and only this 5' hydroxyl up here on the phosphate is not involved in a phosphodiester linkage. So this is why this is called the 5' end of the molecule, and this is called the 3' end of the molecule. And once again, let me reiterate, they don't have to be all pyrimidines. You can have any pyrimidine or purine, any sequence in this that you want. All right, so now you've seen the structure, which is a little intimidating, but let me show you how we're actually going to draw this in class. It's much easier. What we will do is as follows. There's a couple ways to this. First of all, we can simply designate an N, 5', and we can list the bases. I can draw a line here, which represents my sugar phosphate like this. So this is 1/2 of the helix here. This would be the 3' end down here, and I can put any sequence. So A, T, G, G, C, A, whatever. And because I know the two strands, first of all, are anti-parallel, I know the other strand is oriented in the opposite direction. So this is in 3' end, and this is the 5' end down here. And because the bases always occur as complements to each other, A always bonds to T, G always bonds to C, then I know the other sequence would actually be T, A, C, C, G, and T. All right? So this is how we'll draw the double helix. All right. Now, we even abbreviate past there. So what's normally done is turn it sideways and forget about the sugars and phosphate, the backbone, and just worry about the bases. So in this particular case, I could put 5' A, G, T, T, C, A, G, C. And by the way, I'm just making up that sequence because it doesn't make any difference. And I know that the other strand would be 3' T, 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 C, A, G, T, T, C, G, 5'. So this is pretty much how we're gonna be drawing DNA molecules. By the way, one last point here. You will notice that I'm not using any uracils. In DNA, A is bonded to T with 2 hydrogen bonds, which are indicated by these dashed lines. C is bonded to G with 3 hydrogen bonds, the dashed lines here. And of course, it takes more bonds to hold the fat guys together than it does the skinny guys, even though this is no fat talk week. I'm talking about nucleotides, so I think I can get away with that. But what we find is that if we're only dealing with DNA, then we use thymine. When I take a sequence later, as you'll see, when I take a DNA sequence such as, that's Dan, sorry. When I take a DNA sequence such as 5' A, T, T, C, A, G, G, A, T, 3', that when I make a piece of RNA on that, again, it's anti-parallel, and we follow the same base pairing rules, but in RNA, we don't have thymine, it's actually, we have uracil, so we'd have actually 3' UAAG, UCC, UA, 5'. So again, in that particular case, because this part down here is actually RNA, I'm using the uracils instead of the thymines. So really, this will get much easier. It seems pretty complicated at first, but don't worry about it. We'll work it all out.